When we get to the loop of Henle, right, we are going to reabsorb some more water through osmosis as we go down. We're going to reabsorb sodium through symporters as we go up. Also with that, potassium as we go up, and chloride as we go up. So water is typically going to be reabsorbed in the descending limb, and the ions, like sodium, potassium, chloride, are going to be reabsorbed on the ascending limb. We'll also have diffusion to help us with some bicarbonates as well as calcium and magnesium ions. Notice there's no secretion, really. Almost no secretion happens here, just reabsorption. So for us, for this class, we'll simplify it down and just say there is no secretion. There's no notable secretion that occurs in the loop of Henle. It's all reabsorption. It's all the reclamation of things that are still useful. So as we're descending in the loop of Henle, it's not permeable to salt, but it is permeable to water, which is why water is drawn out of the filtrate and into the interstitial space. It's important to note that as we are traveling down into that medulla, that interstitial space is really salty. There's a lot of ions, which is going to use osmosis to draw water out of the filtrate and into that space. So we get about 15% of the filter water that gets reabsorbed, and the deeper we get, the more water that gets removed because the solute concentration gets higher and higher. So the deeper we go down, right, into the medulla, the higher the salt concentration, the more water gets drawn out, right? And then as we go back up, that's when we will reabsorb the ion side of things. We're not worried about water, we're worried about ions. So why is it so salty? Why is this area, like in the medullary region, why does it become salty? Why do we see this difference in solute concentration there? And that's because we are basically, as we ascend the thick portion, we have this salt gradient that is pumping this concentrated sodium into the interstitial fluid. But those thick walls of the ascending portion, not permeable to water. So water cannot move into there. So salt is forced out, but water cannot move in to dilute it. So no osmosis occurs in the ascending portion there. So we're actively transporting, right, using our sodium, chloride, potassium pump, we're actively transporting sodium into this interstitial space. Chloride is following it because it's a negatively charged ion following a positively charged ion, right? Opposites attract. Potassium is going to have leakage channels, so allow it to leak out. So that's going to help us, that's going to leak into the tubular space. So we look what's happening, right? All of these guys are being pumped out, right? Chloride, sodium, and potassium. Sodium continues being actively pumped out. Chloride follows it naturally because negative charge follows the positive charge, whereas potassium has leaky channels that allow it to just flow back into the tubule to be part of the filtrate, the urine. So this is how we create this really salty environment that can draw water out in the descending limb. So a little bit more on this. We have the countercurrent multiplication. This is a mechanism that maintains this gradient, right? So as like when we are reabsorbing that water, that those symporters in the thick ascending limb, again, we're causing that sodium and chloride ions to accumulate in the medulla. This creates a very hypertonic interstitial fluid, right? So it's going to be really, really salty, basically. Urea recycling will also contribute to tonicity, so it will also accumulate in the medulla from the collecting ducts, and then we get this osmotic gradient, basically, where there's a higher osmolarity as you go deeper. So the deeper you go, the saltier it gets, the easier it is for water to be pulled out. So as we go down, that thin descending loop of Henle is going to reabsorb a lot more water. It's going to help to concentrate urine down. With the countercurrent exchange, right, we're doing this exchange between the blood and the vasa recta, which is that one blood vessel that's diving down, kind of following the loop of Henle. Water the solutes in the water being exchanged between the blood there and the interstitial fluid are going in opposite direction. So we have this countercurrent flow, right? So basically the countercurrent exchange will maintain our countercurrent multiplication. Now, we also reabsorb in the early distal convoluted tubule, but there's almost no secretion here. So we're going to continue absorbing without secretion. It's almost like we're just continuing what the loop of Henle was doing in a way. But in this case, right, we're reabsorbing sodium and chloride using those symporters again. So same kind of thing we saw before, where we were pumping sodium chloride in, and then to get into the interstitial fluid, right, we have a sodium-potassium pump, which pumps the sodium into the interstitial space. Chloride follows through those channels. Potassium is now inside the cell. But it's also where parathyroid hormone comes into play. Parathyroid hormone is going to come into play in this section of the distal convoluted tubule to reabsorb calcium. It's also going to inhibit phosphate reabsorption, so phosphate ends up in the urine. So if we look at the early distal convoluted tubule, we're reabsorbing water through osmosis. We are reabsorbing sodium and chloride, and we are reabsorbing calcium as dictated by the presence of PTH. As we go down through the distal convoluted tubule, later on in that, and before we get to the collecting duct, we have a couple different types of cells. We have the principal cells and the intercalated cells. I mentioned these guys before, but just to kind of reiterate them, the principal cells are going to be responsible for the sodium-potassium pumps. This is where we are going to respond to aldosterone. So aldosterone will promote the reabsorption of sodium and therefore water, and the secretion of potassium. We also have ADH receptors here, which will basically put aquaporin 2 channels onto the cell membranes, and so water can reabsorb via these aquaporins. The intercalated discs are just The intercalated cells are just responsible for pH. Again, type A and type B, the way that I would remember them is type A deals with acidic environments, type B deals with basic environments. So if things are too acidic, type A cells will get rid of the hydrogen ions and keep bicarbonate, therefore getting rid of acidic things to get us to more neutral. Type B cells are going to get rid of the bicarbonate, and they're going to reabsorb the hydrogen ions, going from basic to more neutral. And so we kind of get this nice and neat table put together, what's going on. We're going to reabsorb a little bit more water, a little bit more sodium, and bicarbonate, depending on the pH and whatnot. So we're going to be doing this in response to ADH and aldosterone. Urea is just variable, right, if we're recycling it to the nephron loop. And then secretion, we're going to push potassium in. We're also going to push hydrogen ions in to maintain pH balance. The last little piece is the collecting duct. This is where the filtrate can have its last modifications made before excretion down towards the bladder. It is impermeable to salt, but it is permeable to water. So we can get a little bit more water reabsorbed here. This is going to help us really concentrate the urine down. This is going to come down to the hypertonicity of the interstitial space, basically osmosis using those aquaporin 2 channels, again, dictated by ADH. If ADH is there, we have the aquaporin 2 channels, we can reabsorb water. If ADH is inhibited, we do not see those aquaporin 2 channels like that, and we do not reabsorb as much water in the collecting duct. Now, how is this regulated? A lot of this comes down to the various hormones when it comes to the tubule and collecting duct. We have the renin, angiotensin, aldosterone system. Again, angiotensin 2 is going to help us reabsorb water, sodium, chloride, as well as get rid of potassium. Aldosterone will do the same thing, but in the collecting duct. Antidiuretic hormone is going to help us with water. So we have those, again, those aquaporin 2 channels for water that antidiuretic hormone will promote. Parathyroid hormone will promote reabsorbing calcium. And then atrial natriuretic peptide, this is the one that is going to help push things out. It's going to secrete things. It's going to help secrete things like sodium and the chloride ion. So it's going to help us like get rid of those things, which means water will also go with it. So we will not reabsorb as much water either with ANP. ANP again is released when when blood pressure is like high, when blood volume is high, because if we get rid of like the sodium and we get rid of some water, we can lower blood pressure, we can lower blood volume. Now, how do we balance all these electrolytes? Because what we get in our diet varies, right? We don't eat the exact same thing at the exact same time every single day. There is variation in the amount of electrolytes in the different foods we eat. And so our kidneys have to work really hard to try to balance these electrolytes, to make sure that no matter what we've eaten and no matter what has come in, that we are maintaining these electrolytes in a very specific amount. And so 90% of the sodium and potassium is reabsorbed super early, and that just goes right back into the bloodstream to help regulate that. And this is if there's no regulation. So if everything's like fine and the levels of these two are not changing much, 90% of them go back into the bloodstream. Aldosterone will control another 10% of that. And if we have a rise in blood potassium, that will trigger aldosterone from the adrenal glands. If sodium falls and blood volume falls, that's where aldosterone kicks in via the renin-angiotensin-aldosterone system. Because those juxtaglomerular cells will pick up on that. They'll pick up on that loss of blood volume, they'll secrete the renin, which then angiotensin 1, angiotensin 2, aldosterone. If we have too much sodium in water, right, this is where we get the ANP, right, as well as the JGA to inhibit renin. So we can say, no, we don't need renin now. We don't need to, promote the reabsorption of salt. We don't need to promote the reabsorption of water. And so we see a decrease in those things in the blood. Now, because we're reabsorbing sodium, that can also stimulate the secretion of other positive ions because, if we're reabsorbing something with a positive charge, we need to get rid of something with a positive charge to make sure that the pH isn't changing and the electrochemical gradient's not changing significantly. And so we can experience some problems if we are completely out of whack. So acidosis, this is basically where we have to get rid of a bunch of hydrogen ions and we have to keep potassium. So acidosis will do that. We're going to get rid of hydrogen instead of potassium. But because of that, because we're so focused on maintaining pH, we can end up with what's called hyperkalemia. Hyperkalemia is too high of a level of potassium. Alkalosis, this is where we have too much secretion and excretion of potassium, and this can lead to hypokalemia. And so hyperkalemia will also promote secretion of potassium and inhibit the secretion of hydrogen ions, and that can lead to acidosis. So you can see how hyperkalemia and acidosis can be kind of a bit of a vicious cycle. So when it comes to maintaining blood pH, we reabsorb that bicarbonate, we get rid of the hydrogen ions, which means urine is going to be a little bit more acidic than blood. Blood's about a 7.4 pH. We're going to see under neutral with the pH of urine because the bicarbonate can't just directly pass through the tubule membrane. It has to be converted to carbon dioxide and water using our carbonic anhydrase enzyme. Carbon dioxide totally can cross in, where then we can reverse. So it's kind of weird if we look at like the picture here. We see that we have to use our carbonic anhydrase to break it down. Water and carbon dioxide can then move, and then we rebuild it back and then excrete it like further. So a little bit strange. We have like a couple extra steps because the bicarbonate can't directly just pass through. And then with the proximal convoluted tubule, we're going to use sodium hydrogen pumps to get sodium out and hydrogen in. Again, we will compensate if we have some metabolic pH problems like alkalosis and acidosis. If we are alkalosis, we're going to get rid of more bicarbonate to try to compensate, and if we're experiencing acidosis, we will make more bicarbonate, basically using glutamine to just make more bicarbonate to push that into the bloodstream to try to compensate. Now, diuretics are going to help control blood pressure and relieve things like edema, basically by getting rid of extra fluid. So you're going to increase your urine volume and decrease blood volume with a diuretic. So diuretics, in essence, do make you pee. Different diuretics will work a little bit differently. They'll work on different locations of where water is reclaimed. So remember, water is reabsorbed almost everywhere along this nephron. So we have loop diuretics. They're going to inhibit sodium transport in the loop of Henle. This can inhibit up to 25% of your water reabsorption. So these are very powerful. So you're going to end up with a lot of water being left in the urine. Thiazide diuretics, these will inhibit sodium transport in the distal convoluted tubule. So this is going to be a little less powerful. Carbonic anhydrase inhibitors, these are weaker. They're going to inhibit water reabsorption when bicarbonate's reabsorbed. And we also will get rid of bicarbonate. And then potassium sparing diuretics, this is going to block aldosterone receptors so that we get rid of sodium and we keep potassium. And so the way that this one, why this one's a diuretic is if we get rid of the sodium, water is going to go with it too. So like spironolactone is going to be an example of that one. And some of these are used for things like blood pressure and edema, but other things are used for other things. I know like spironolactone is actually something some people will use for things like acne. Now, dilute urine, this is going to be, remember if we talked about cortical versus juxtamedullary nephrons, remember dilute urine's coming from the cortical nephrons. It's going to be really similar in terms of osmolarity to blood. So this is when you don't need a lot of ADH, your ADH level's low, a person's is well hydrated basically. So we can produce dilute urine because we have an excess of water. So we do reabsorb solutes, right? But water permeability is low, so water doesn't follow because the water's already there. And so the fluid going into the distal convoluted tubule is more dilute. Then because of that, we're not going to be very permeable to water. We're not going to be regulated by ADH here. It's going to remain impermeable to water. And so like basically if ADH levels are low, we're going to do some reabsorbing early on, but then as we go down through the loop of Henle and into the DCT and collecting duct, we're not going to be reabsorbing as much water. So the urine ends up very dilute, right? It ends up very, very dilute. So this is like when, like again, these ones are going to be kicking on hardcore when you're well hydrated. You drink water and your ADH levels are not kicked on super high because there's no need for you to create super, super dilute urine. But the juxtamedullary nephrons with these super long loops, this is where we are going to kind of focus to produce concentrated urine when we don't have enough water in our system. So we have a higher level of ADH. The collecting ducts are very permeable for water via those aquaporin 2 channels so that we can reabsorb more water. And so the fluid becomes very concentrated. The solutes in the fluid become very concentrated. This is also going to carry urea into the medulla, which is going to contribute to the osmolarity. And we see we went from like about 100 to 1200. So 12 times higher in terms of concentration in comparison to when we're using those cortical nephrons.